Welcome to Euclide Academy. Today we're going to solve the 2020 Biology Paper 2. We're going to look at this past paper and we're going to revise questions from this from the past paper. So this is for internal candidates, and the instructions are that no phones are allowed in the examination room, and you're advised to spend no longer than 1 hour on section A and no longer than 45 minutes on section B. So that means that the entire paper, that is section A and section B, you have got 1 hour 45 minutes, and how you time or how you put yourself in a position to answer all the questions within the specified time will really determine how far you're going to answer the questions. So all in all, you have to time yourself to make sure you are within the stipulated or the given amount of time. So let's get into it. We have got question 1 from section A, which says answer all questions in the spaces provided in the question paper. So table 1 shows cells and tissues found in living things. So we've got a table here, and they've given us cells on top: red blood cell, root hair cell, palisade cell, and the sperm cell. And then we've got tissues. We've got the xylem tissue, the nerve tissue, the epidermal tissue, and the muscle tissue. Question A reads, from table 1, identify a plant cell, an animal cell, a tissue whose function is to cover and protect plants from. Okay, so we're going to look at the first two questions for now. So identify a plant cell. This probably will be much easier, I believe. So a plant cell, we know from cells this will be root hair cell, right? So you can say the root hair cell, or you can put the palisade cell. Palisade cell is also plant cell. So any of these two options. An animal cell, you can either put the, of course, red blood cell, or the sperm cell. These are the two animal cells we have. So 1 mark for each. A tissue whose function is to cover and protect plants from mechanical damage and infections. So if we look back at the table, we have got tissues down here. So we are not going to look at this row because we are not talking about cells, we're talking about tissues. So a tissue whose function is to cover and protect plants from mechanical damage. So xylem tissue is doesn't cover the plant, it protects. It is inside, which acts it's part of it deals with transportation of we'll talk about this probably in the lesson. We did talk about this. I'm sure you can watch the video on transport. We talk about the function of xylem tissue. The nerve tissue, no, it's not even a plant tissue. The muscle tissue is not even a plant tissue. So we've got the epidermal tissue, so one which covers and protects. So this will be the correct answer for question number 1A3. So 1A3 will be epidermal tissue. That will be the answer for this one, which will be the epidermal tissue. Then we have question. Okay, so this one's epidermal tissue. A tissue whose function is to transport water and dissolved mineral salts in plants. Okay, I was just mentioning about this transportation of the xylem tissue. So definitely this is, when we go back to the table, we can see the xylem tissue here is the one which is involved in transportation of mineral salts from the roots to parts of the plants, like getting water and all or the dissolved minerals. So we're talking about the xylem tissue here. The xylem tissue, a cell found in the reproductive system. That would be fairly easy. A cell found in the reproductive system. So we already know sperm cell, very much involved in reproduction. So this would be sperm cell. In which tissue are the following cells found? Red blood cell, that is palisade cell. So the red blood cell, this will be a blood tissue. That's a blood tissue. Then the palisade cell. So here we're talking about the cells which the palisade cells are found mostly in the leaf of a plant, on leaves, and we're talking about the palisade layer or palisade tissue. So you can say the palisade layer, or if you want you can say the palisade tissue, palisade layer or palisade tissue, and this is where we find the palisade cells. So name one organ found in a plant. Okay, so we can talk about the stem, the leaf, flower. We can talk about fruit, roots. Okay, so there are many organs that are found in the flower. Just need one on this one. Then the last question in part 1, in question 1, says an animal cell. Name one organ found in an animal. An organ found in an animal. You can mention the eye, the ear, the lung, heart, the blood vessels. You can mention the aorta, and the tongue. Lots and lots of organs you can mention. Okay, so the stomach. So these are organs you can mention. So anyway, I think that is where we end on question 1. So let's jump to question 2. Question 2 reads, Table 2 shows the concentration of various substances in blood plasma, glomerular filtrate, and urine. Name the process responsible for the formation of glomerular filtrate. Okay, first of all, we need to study the table. We've got the substance and the concentration here. So the substances that we have, we've got urea here, glucose, salt, and protein. Then blood plasma, glomerular filtrate, and urine. And then we've got the different concentrations inside here according to the fluid or the substance. So in blood plasma, urea, it has this concentration, and glomerular filtrate this concentration, urine that concentration, glucose, etc, etc. So question A1 says, name the process responsible for the formation of glomerular filtrate. So what is glomerular filtrate anyway? So in the lesson, if you're part of the class for excretion, this topic definitely has to do with excretion. We didn't mention, we explained for you what glomerular filtrate is, and I think this lesson is all done and complete. Then you can easily answer the this question if you've been part of the lesson. So if you haven't registered yet for the lessons, I would encourage you to do that. It will be really helpful for you, and you'll be on your way to get those great marks. So the process responsible for the formation of glomerular filtrate. Now glomerular filtrate, this is like it's a substance which a liquid or fluid which comes about as a result of filtration in the kidneys, and there's a process which is responsible for that, for filtering. As you can see, glomerular filtrate is a result of filtration, and the process is called ultimately as ultrafiltration. Ultrafiltration. So what is ultrafiltration? So in the biology class, when you register for lessons, we completely answer for you this topic, ultrafiltration. We define for you and we talk about how it comes about and most of the issues involved with ultrafiltration. So anyway, the process responsible for the formation of glomerular filtrate in the kidneys is ultrafiltration. Which of the substances in table 2 above does not pass through the glomerulus during filtration? Okay, so which one does not pass through? So if you can see, this is blood. So the way things move, it's like in the kidney, when we talk about excretion, that is the removal of the unwanted substances in the body, blood. Okay, then there's the formation of glomerular filtrate, then there's urine. Okay, so it's like this is the steps. So from blood you can see there's urea 0.04, glucose 0.1, salt 0.46, and protein 8.06. Then there is some filtration which goes on to give us glomerular filtrate, and then you see urea remains the same, glucose and then salt they kind of remain the same concentration. And then we can see here that the protein has reduced, like there was something which happened here for it to go from 8.0 to 0.0, right? So which of the substances in table 2 does not pass through the glomerulus during filtration? So definitely this has been filtered off, it does not pass through. The rest you can see they've passed through. There's this, there's that. So protein does not pass through. So the answer here will be protein. And why does protein why doesn't it pass through is because of the size and that still needed in the body, and we do mention a lot of that in the biology lesson on filtration. I think we talked about all the reasons. I can mention enough of that. Next part of the question says give a reason for your answer in A2 above. So A2 here, let us give a reason. Why do we say protein? Because proteins are large because of the size of the proteins. Proteins are too large to pass through the filtration barrier. They're filtered off because proteins are large molecules. Okay, because of the protein size, or they are too large to pass through, hence they are filtered off. Okay, that's the reason why. Explain why the concentration of urea in the glomerular filtrate is much less than the concentration of urea in urine. Okay, let's check out the question again. Explain why the concentration of urea in the glomerular filtrate. Okay, so let's look at the table. So this is a glomerular filtrate. Concentration of urea is 0.04. Now they're saying like why is it much less than the concentration of urea in urine, which is 0.02, sorry, 2.0. So why this difference? Why do you have 0.04 here and 2.0 despite being in the filtrate, you know, then going to urine? What really happens that the concentration here now increases? So when you talk about concentration, if you understand concentration, really we're talking about the amount of solute per decimeter, or per volume. Let me just say per volume, the amount of per volume of a solution, that is concentration. So if you've got more solute per volume of a solution, then that is, you know, mean the concentration is high. So here, why do we have 2.0 in the glomerular filtrate and 0.04 in the urine and then 0.04 in glomerular filtrate? Well, it's because as this we talk about filtration in class, as we have the glomerular filtrate here, as it's being filtered off. there is what we call selective reabsorption, which some useful substances like glucose, water, and amino acids, they are reabsorbed back into the blood, which makes the concentration of urea to be more. So if I were to illustrate for you, so let's say this is what we have here. We have different substances in here. You have water molecules, you have urea, and then what else? You have amino acids, okay, and then so water, urea, amino acids, and we have glucose. Okay, we have glucose there. So during selective reabsorption, some of these are taken. So the concentration of urea in here, maybe it could be 0.04. okay, if we take into account the other substances or the other molecules, let's say about 1 2 3 4 5 6 7 8 of them, so the concentration is 0.04, meaning urea occupies as not much space compared to, because there are more people in here, so to say. I just want to make it so that you understand, because there are more molecules occupying this room. Now if we reabsorb, selective reabsorption is reabsorbing some of these molecules, so we can have, maybe we have water taken out, then we have glucose again reabsorbed, and then we have amino acids again reabsorbed. So you can see that now we remain with only 2 molecules of urea, and then this space here is even too large, so this space will be kind of reduced, because it only has to accommodate 2 molecules now. So we see that the concentration now of urea per volume will be much higher compared to this part, because here it had some other molecules in it. But now since these have been reabsorbed back into the bloodstream, then we've got a high concentration of urea. So when we talk about why is it the concentration of urea in the filtrate is less, in the glomerular filtrate is less compared to the concentration of urea in urine, it's because of selective reabsorption. Selective reabsorption, which reabsorbs substances, some useful substances, back into the blood. Hence we have got urea is not reabsorbed, hence the concentration of urea gets high or becomes more. Outline the internal structure of a kidney. So talk about the internal structure of a kidney. I think they can outline the internal structure of a kidney. So if it was drawing, we would have drawn, but it's about outlining. So in the kidney we will have the veins. We can talk about the veins. I'll just put it in point form. If you want, you can expand further on that. So we can talk about the renal got the renal vein, that is one structure, and then we could talk about the renal artery and renal artery, and then we can talk about the pelvis, and then we can talk about the medulla, and we can talk about the cortex, and then we have got the nephrons somewhere in between, somewhere in these parts, the nephrons. If you want, you can include the nephrons. So these are the things we talk about when you mention the internal structure of a kidney. So you can mention these, as you can see, there are about 3, 4, 5, 6 parts you can talk about in the kidney, and then you just have 4 marks here. Definitely you should be able to score those 4 marks. So that is for question 2, and we can go ahead and do the next question. Question 3 which show figure 3 shows stages in the life cycle of a housefly. So this is the life cycle of a housefly. Identify stages P and R. So for a housefly, you know the life cycle, that the stages we've got this P, these are eggs. This is the egg stage. When we say eggs, then this is the larva, also known as maggots. And then we have the pupa, and then the adult housefly. So identify stages P and R. So P, of course, those are the eggs, or the egg stage, eggs. Then R, we have the pupa. Okay, so those are the stages for housefly. Let's look at the next questions. Which stage is useful in the decomposition of matter? Which stage is useful in the decomposition of matter? So we know that houseflies, they lay eggs on dead bodies of organisms or decaying matter, feces, or any rotten thing, and then the eggs, they become the larva, then they start feeding on the garbage or the dead bodies. So the stage useful in the decomposition of matter, that is the larva stage, or the maggots. And then we have the which stage is involved in the spread of diseases. So for a housefly, that will be the adult stage, is the one which is involved in the spread of diseases, because they they'll sit on garbage, pick up some pathogens, and then carry the pathogens on their mouth, legs, and spread them to the surfaces and food stuff, and people consume or put their hand in the mouth. After touching the surface, they get infected. So that is with the housefly. So which stage is involved in the spread of disease? That would be S, or the adult stage. So we'll say stage S. Which two diseases in which a housefly is a vector? So what is a vector? So in the biology class, we'll define what the vector is. That is an organism that does not cause a disease itself, but just spreads a disease by carrying the pathogen that causes the infection. So with a housefly, we've got cholera, it's one of the diseases, then typhoid, or dysentery. Explain how mosquito is similar to a housefly in terms of life cycles. So life cycle of a mosquito and housefly, they quite similar, because they both start as eggs, and then they both go through the larva stage. But only in a housefly, it's now in a mosquito is known as a wriggler, not a maggot. Egg, larva, then the pupa stage, and finally the adult stage. So that is how the mosquito is similar to housefly in terms of the life cycles. The spread of diseases, they are both vectors. meaning they don't cause a disease, they just carry the pathogens, and they both do it in their adult stage. So there are similarities between the life cycle housefly and mosquito in terms of spreading diseases. They are both vectors and they both spread the diseases in the adult stage. Then that is enough to score 1 mark for each. Explain one way by which a housefly can lead to the spread of cholera. Explain one way by which a housefly can lead to spread of cholera. So a housefly can settle on garbage, and then it picks up the pathogen, and then it goes and settles on open food, or food which has been left open, and then a person comes and eats food which has been left on the open for a long time where the housefly had settled, and the person gets the pathogen into their body and they get infected. So how can you tell this story to get 1 mark? So explain one way by which a housefly can lead to the spread of cholera. By spreading the cholera, the pathogen, from infected area, or infected by settling on infected or from garbage, or person's stool or vomit who's been a cholera victim, settling on those, and then taking the pathogen on food, a food substance, on food which is then eaten by a person. Okay, it's more of a story by another host or another individual. Okay, and then that individual gets infected. So I think you get the story. You can literally tell what's going on. So that is how a person can one way which a housefly can lead to the spread of cholera. Okay, so it settles on infected person's vomit or stool, and then gets the pathogen, carries the pathogen, legs, mouth, and then settles on food. That food is eaten by the individual, so that person gets the cholera. So that is with question 3. And by the way, I think I can mention a recommended book for biology which I use and I recommend it to most of my students, so I'll put it somewhere here. So this is a recommended book. You can get this currently. It is delivered to you when you purchase this book, so you can call the number on the screen to buy this biology book. Get yourself the biology book. I'll literally recommend this. It's a very good book, and it will definitely help you, as it has everything from grade 10 to grade 12, and most of the things, almost everything, 99% of the things I use when answering these videos, definitely, and when teaching, when my students usually use this book. It's a pretty amazing book. Alright, so let's go to question 4. Figure 4 shows structures of 2 neurons. So I've got neuron 1 and neuron 2. So these are neurons. Identify 2 neurons shown. So this is on sensory organs or nervous system. This is where the topic is coming from. The question is coming from that topic, and we definitely did this lesson as well in class, so for the biology, and we mentioned that the different types of neurons that we have. So these diagrams are showing the different types of neurons. So neuron number 1, identify the 2 neurons shown in figure 4. Neuron 1 is what we call the motor neuron.